FSO ഫുഡ് സേഫ്റ്റി ഓഫീസറിൻ്റെ കുറച്ച് MCQ ക്വസ്റ്റ്യൻസ് ആണ് ഇവിടെ ഡീൽ ചെയ്തിരിക്കുന്നത്. മൈക്രോസ്കോപ്പി, സ്റ്റെയിനിങ് ആൻഡ് കൾച്ചർ ടെക്നിക്സിൻ്റെ ക്വസ്റ്റ്യൻസിലാണ് നമ്മൾ സ്റ്റാർട്ട് ചെയ്യുന്നത്. വിച്ച് ടൈപ്പ് ഓഫ് മൈക്രോസ്കോപ്പി പ്രൊവൈഡ്സ് ദ ഹയസ്റ്റ് റെസൊല്യൂഷൻ? ഓപ്ഷൻ എ ലൈറ്റ് മൈക്രോസ്കോപ്പി, ഓപ്ഷൻ ബി ഇലക്ട്രോൺ മൈക്രോസ്കോപ്പി, ഓപ്ഷൻ സി ഫ്ലൂറസെൻസ് മൈക്രോസ്കോപ്പി, ഓപ്ഷൻ ഡി ഫേസ് കോൺട്രാസ്റ്റ് മൈക്രോസ്കോപ്പി. Answer option B electron microscopy. Explanation: electron microscopy uses electron beams for imaging, providing higher resolution than light microscopy. Next question: which staining technique is used to differentiate bacterial species into gram positive and gram negative? Option A acid fast stain, option B gram stain, option C symbol stain, option D negative stain. Answer option B gram stain. The gram stain differentiates bacteria based on the structure of their cell walls. Next question: what is the primary purpose of using a streak plate technique? Option A to quantify bacteria, option B to isolate pure colonies, option C to preserve bacteria, option D to identify motility. Answer option B to isolate pure colonies. The streak plate technique helps to separate individual bacteria species. Next questions based on sterilization techniques. Which of the following methods is the most effective for sterilizing heat sensitive materials? Option A autoclaving, option B dry heat sterilization, option C filtration, option D incineration. Answer option C filtration. Filtration is used for sterilizing heat-sensitive liquids and gases without exposing them to high temperatures. Next question: what is the principle behind autoclaving? Option A radiation, option B moist heat, option C dry heat, option D chemical stimulants. Answer option B moist heat. Autoclaving uses steam under pressure to kill microorganisms by denaturing their proteins. Next question: what is the term for the time required to kill all microorganisms at a specific temperature? Option A D value, option B Z value, option C thermal death time, option D thermal death point. Answer option C thermal death time. Thermal death time is the time needed to kill all microbes at a set temperature. Next, culture media topic related questions. Which type of culture media is used to grow fastidious organisms? Option A selective media, option B differential media, option C enriched media, option D minimal media. Answer option C enriched media. Enriched media contain additional nutrients to support the growth of fastidious organisms. Next question: McConaughey agar is an example of which type of media? Option A selective and differential media, option B enriched media, option C minimal media, option D transport media. Answer option A selective and differential. McConaughey agar differentiates lactose fermenters and inhibits gram positive bacteria. Next questions based on factors influencing microbial growth. First question: which of the following factors does not influence microbial growth? Option A temperature, option B light, option C pH, option D humidity. Answer option B light. While light affects some microbes, it is not a universal factor influencing microbial growth. എപ്പോഴും ക്വസ്റ്റ്യൻ നന്നായിട്ട് ശ്രദ്ധിക്കണം. ഇതിൽ ഇപ്പം ഈ ക്വസ്റ്റ്യനിൽ തന്നെ ഏതൊക്കെയാണ് ഫാക്ടേഴ്സ് എന്നല്ല ചോദിച്ചിരിക്കുന്നത്, ഏതാണ് അതിലല്ലാത്ത യൂണിവേഴ്സൽ ആയിട്ടുള്ള ഫാക്ടർ അല്ലാത്തത് എന്നാണ് നമ്മൾ സെലക്ട് ചെയ്യേണ്ടത്. അതായത് മോസ്റ്റ് ഓപ്ഷനിൽ നിന്ന് മോസ്റ്റ് അപ്രോപ്രിയേറ്റ് ആയിട്ടുള്ള ആൻസറാണ് നമ്മളെപ്പോഴും ചൂസ് ചെയ്യേണ്ടത്. അപ്പോൾ അത് നമ്മളെപ്പോഴും ക്വസ്റ്റ്യൻസ് വരുമ്പം ഓരോ ക്വസ്റ്റ്യൻസും ഇമ്പോർട്ടൻ്റ് ആയിട്ടുള്ള ഒരു ക്വസ്റ്റ്യൻ വായിക്കുമ്പോൾ തന്നെ അതിൽ ഇമ്പോർട്ടൻ്റ് എന്ന് തോന്നുന്നത് ഇപ്പം ഇതിൽ നിന്ന് ഏതാണ് അല്ലാത്തത് എന്നൊക്കെ ചോദിക്കുന്ന ക്വസ്റ്റ്യൻസിൽ അപ്പോഴേ അത് അണ്ടർലൈൻ ചെയ്ത് വെക്കുക ആ ക്വസ്റ്റ്യനിൽ. എന്നിട്ട് വേണം നമ്മൾ ആൻസർ ചെയ്യാൻ, അല്ലെങ്കിൽ നമുക്ക് ഒരുപാട് മിസ്റ്റേക്ക് വരാനുള്ള ചാൻസ് ഉണ്ട്. സോ കൂടുതൽ ഫോക്കസ് ചെയ്ത് ആൻസേഴ്സ് എഴുതാൻ ശ്രമിക്കണം. Psychrophiles grow best at which temperature range? Option A 0 to 15 degrees Celsius, Option B 20 to 30 degrees Celsius. Option C 37 degrees Celsius. Option D 50 to 60. Answer option A 0 to 15 degrees Celsius. Psychrophiles thrive in cold environments, typically between 0 to 15 degrees Celsius. Next questions based on growth curve. In which phase of the bacteria growth curve do cells divide at a constant rate? Option A lag phase, option B log phase, option C stationary phase, option D death phase. Answer option B log phase. During the log phase, bacteria multiply at an exponential rate. Next question. Which phase of the growth curve is characterized by the depletion of nutrients and accumulation of waste products? Option A lag phase, option B log phase, option C stationary phase, option D death phase. Answer option C stationary phase. Stationary phase occurs when growth rate slows due to the limited resources and waste accumulation. Next questions based on thermal death time and thermal death points. Thermal death point is defined as option A the lowest temperature at which all organisms are killed in 10 minutes. That option B the time required to kill all microbes at a given temperature. Option C the temperature required to kill a specific microorganisms. Option D the time required to reduce the microbial population by 90%. Answer option A the lowest temperature at which all microorganisms are killed in 10 minutes. Thermal death point indicates the minimum temperature for microbial death in a specified time. What is D value in microbiology? Option A time taken to reduce the population by 90% at a specific temperature. Option B the temperature required to kill all microbes. Option C time to kill all microbes at a given temperature. Option D change in temperature required to reduce D value. Answer option A the time taken to reduce the population by 90% at a specific temperature. D value represents the time needed to achieve a 1 log reduction in microbial population at a specific temperature. Next question. Z value represents option A the time taken to kill all organisms at a specific temperature. The temperature option B the temperature change required to change the D value by 1 log. Option C the temperature at which all organisms are killed in 10 minutes. Option D the time required to reduce microbial load by 90%. Answer option B the temperature change required to change the D value by 1 log. Z value quantifies the thermal resistance of a microorganism. Further some additional questions. What is the function of a mordant in staining procedures? Option A increase the size of cells. Option B fix the dye to the specimen. Option C decolorize cells. Option D counter stain the cells. Answer option B fix the dye to the specimen. A mordant enhances the binding of the dye to the cellular components. Next question. Which of the following sterilization methods uses ionizing radiation? Option A UV light. Option B gamma rays. Option C boiling. Option D pasteurization. Answer option B gamma rays. Gamma rays are a form of ionizing radiation used for sterilization. Next question. Which microbial growth phase involves adaptation with no immediate increase in cell number? Option A lag phase. Option B log phase. Option C stationary phase. Option D death phase. Answer option A lag phase. In the lag phase, bacteria adapt to the new environment before starting division. Next question. Which staining method is primarily used for Mycobacterium tuberculosis? Option A gram stain. Option B acid fast stain. Option C endospore stain. Option D capsule stain. Answer option B acid fast stain. Explanation. The acid fast stain is used for Mycobacterium species due to their waxy cell walls. Next question. Which sterilization method is most suitable for sterilizing glassware? Option A autoclaving. Option B filtration. Option C dry heat. Option D ethylene oxide. Answer option C dry heat. Dry heat sterilization is effective for glassware, destroying microbial cells and spores. Next question. Which of the following is a chemically defined medium? Option A nutrient agar, option B McConkie agar, option C glucose salt agar, option D blood agar. Answer option C glucose salt broth. A chemically defined medium has a non-chemical composition. Next question. In what phase of microbial growth is the rate of cell death equal to the rate of cell division? Option A lag phase, option B log phase, option C stationary phase, option D death phase. Answer option C stationary phase. In the stationary phase, cell growth and cell death rates balance out. Next question. Which factor does not affect thermal death time? Option A pH, option B initial microbial load, option C temperature, option D light intensity. Answer option D light intensity. Thermal death time is affected by temperature, pH and initial microbial load but not light intensity. Next question. What is the term for bacteria that require high levels of salt for growth? Option A halophiles, option B psychrophiles, option B thermophiles, option D acidophiles. Answer option A halophiles. Halophiles thrive in environments with high salt concentration. Next question. What is the significance of the Z value in food safety? Option A determines the lethal dose of radiation. Option B measures the effectiveness of a sterilization process. Option C indicates the temperature change needed for a tenfold change in the D value. Option D measures the pH tolerance of microbes. Answer option C indicates the temperature change needed for a tenfold change in the D value. The Z value helps in designing effective thermal processing schedules for food safety. Thank you.